LIMS Nucleus by Laboratory Automation Solutions. Contact us at info@labsolutions.com or visit our website at labsolutions.com. Laboratory information management systems come in two general configurations, monoliths or systems. With a monolith, raw data and experimental design are entered and a single software application handles all processing, outputting final results which might include data loaded into a database, graphical analysis, or reports. Monoliths are great when you have a stable process that is not changing. You can take advantage of full automation, minimizing the FTE requirements, and enjoy a consistent and reproducible process. All enhancements, bug fixes, and upgrades are outsourced to the vendor. Quite often, user groups provide resources for problem solving. Another approach to LIMS development is the systems approach. With systems, the activity of multiple independent pieces of software are integrated to produce the final workflow. Each piece of software provides limited functionality, may be upgraded on its own schedule, and modified or replaced as the process evolves. Individual components might be commercial, open source, in-house developed, or proprietary but contracted out to a software vendor. A systems approach is much more amenable to a research lab where workflows are constantly evolving. LIMS Nucleus fits into the systems approach of LIMS development. LIMS Nucleus has a limited set of features including cataloging multi-well plates, reformatting plates, tracking samples, generating robot control files, applying assay data to sample plates, identifying hits, and rearraying. A goal of LIMS Nucleus is to avoid feature creep. New features are incorporated by adding new modules. This slide provides a schematic of the canonical workflow handled by LIMS Nucleus. A plate set of samples is processed through an assay, generating data. The data is applied to the plate set to identify wells that contain hits. Once hits are identified, a robot control file is generated that allows for the rearray of hits into smaller collection of plates. The smaller plate set can then be reassayed for confirmation or moved on to a secondary assay. At any stage, it may be desirable to rearray the plates into a higher density prior to assay. LIMS Nucleus works with a series of nested entities. The highest level entity, a project, contains plate sets which contain plates which contain wells which may hold samples. An assay run may be associated with a plate set. A layout is applied to a plate set. A collection of hits is associated with a project, and a sample can be applied to a target. Higher level entities, such as project, plate set, and plate, have dedicated pages in the LIMS Nucleus interface that contain a tool icon that provides the various functionality that can be applied to each entity. LIMS Nucleus can work with a variety of different assay types. A few examples are displayed on this slide. Samples in the system can represent antibodies, small molecules, or siRNAs. Targets can be the associated antigens, enzymes, and cells. The goal of LIMS Nucleus is to track samples through multi-well plates and identify samples that are hits in assays. Although targets are not the focus of LIMS Nucleus, target information can be critical to the proper evaluation of an assay. Consider an analysis where an antibody is being screened against antigens that represent the human, murine, and monkey homologs of an antigen. LIMS Nucleus will catalog and export target information for merging with the auxiliary software. Sample types are identified by using layouts. LIMS Nucleus provides over 40 built-in layouts that primarily cluster controls in column 12 of a 96 well plate or columns 23 and 24 of a 384 well plate. Users can design and import custom layouts distributing controls anywhere on the plate. Replicates are also possible. More information is available at labsolutions.com under Layouts. Once controls have been identified, algorithms can be implemented to identify hits. First, Responses are background subtracted and then normalized either to the highest signal on the plate or to the mean of the positive control. In a binding assay, a threshold can be set at the mean of the negative controls plus or minus 2 or 3 standard deviation units. Anything above the threshold is a hit. For assays similar to affinity optimization, the mean of the positive controls, which would be the parental antibody, is used as a threshold, and anything with improved binding above this mean would be considered a hit. A third option that is not displayed on this graphic is the top N hits. For example, if you have a low throughput secondary assay that can analyze only one 96 well plate, you may want the top 90 hits with 6 controls. Details of the calculations performed are explained under Algorithms on labsolutions.com. Once hits are identified, a robot control file is generated that allows for the rearray of hits into a smaller collection of plates. Additional features of LIMS Nucleus include import sample accession IDs, import target IDs, import barcode IDs, and import custom layouts. The utility of sample and target accession IDs can be seen in Tutorial 4. Scroll to the bottom and look at the results of exporting assay data. If accession IDs are utilized, they will appear in the export table as they do here for target accessions. This provides you with the opportunity to merge LIMS Nucleus data with data from other components of your LIMS system. If you have questions as to the suitability of LIMS Nucleus for your process, contact us at info@labsolutions.com.